So as Rob already said, I'm a postdoc from the University of Ljubljana, and I will talk about a specific subtype of temperate phages today, which are called phage regulatory switches. So as most of you know, phages usually follow two lifestyles, either lytic or a lysogenic cycle, resulting either in the lysis of bacteria or in the integration of the phage genome into bacterial genome. But actually, there are also alternative bacteriophage lifestyles, such as active lysogeny. Sorry. Okay, sorry. This is basically a lifestyle where the phage also integrates its DNA into the bacteria, but by this it interrupts a specific gene, so it inactivates this gene. And what happens after is that when bacteria is in need of this gene, this phage is actually able to regulate its excision, making the gene again active, and then this can either be reversible, so the phage can get reintegrated and the gene inactivated, or the phage can be lost and the gene is active, like forever or until the cell dies. This was very nicely summarized in this review paper by the group from Tel Aviv, where they managed to really summarize this kind of phage regulatory switches, which are found in many different bacteria. So we are working on Bacillus subtilis. So this is a gram-positive bacteria, which is one of the best studied ones. Specifically, we work on P9B1 isolate, which was isolated from soil. And this is also a sporulating bacterium, and this will be of importance today because I will also talk a bit about sporulation. So just a brief introduction of sporulation and how it happens. So basically, when the environmental conditions become scarce, there is asymmetric division, which results in the mother cell and the spore formation. And then this spore undergoes engulfment, making separated spore. This then maturation follows, and after the cell death of the mother cell, the spore is dormant in the environment and is resistant to many different stresses. So we are also studying, besides of Bacillus subtilis, we also study regulatory switch phage SP beta, which is best studied phage of that subtype. It's quite big, so 134 kilobase pairs. And what is important, it is usually located inside the SPSM gene of Bacillus subtilis. SPSM is involved in spore coat formation during sporulation. And then two other genes are of importance today because I will mention them again. SPRA is the integrase, so helps to integrate into the gene. And SPRB is excisionase involved into the excision of the phage. So basically what was shown before was that when at the onset of the sporulation process, the excision of the SP beta phage happens only in the mother cell. So then we have a complete SPSM gene, which is expressed, and it can produce a normal spore coat. However, in the pre-spore and in the final spore, the phage is still integrated. So basically, the phage is replicated together with bacteria and is kept in the cell, is not lost. So what we want us to do is to follow this process happening. So we started first at the population level. So we used first just a PCR method. So here I'm showing integrated phage and excised phage. So basically when we have the phage integrated, we have specifically amplifications for ATTR. And when we have excised phage, we have ATTP or ATTB sites amplified. So you can see that while the Bacillus subtilis is growing, and here you can see a typical growth curve where the stationary phase starts around 8 hours. So you can see that at the onset of stationary phase, we can also start to see appearing the amplifications for ATTP or ATTB. So this is when the phage starts excising. However, the signal for the integrated phage is present kind of all the time. So it seems that a part of population keeps the phage integrated. And to get a bit more quantitative information into this, we also performed qPCR, where we saw then that at 8 hours, the signal for the excised phage starts appearing and then kind of reaches its maximum at 16 hours. And this is until the observed phenotype was monitored the last time at 48 hours. So it reaches max around 5%. So we were wondering what happens with these cells at a single cell level, because it seems there is some heterogeneity, some cells excise the phage and some don't. So we decided to do some fluorescent reporters. So here is the first one we designed. So this is the transcriptional fusion with attachment gene SPSM. So upon excision, this SPSM gene becomes complete again and then can be expressed. So if you remember from before, this happens only in the mother cell, not in the tree spore. And this is what we observed. So here you can nicely see the spore, and then you can see the fluorescent signal localized really in the mother cell. The second reporter now I constructed was the transcriptional fusion with SPRB, so the excisionase. Here it's expected that this is expressed just before the excision, and post-excision the SPRB is not expressed anymore. And similarly, we also obtained signal which was localized only in the mother cell, so where we expect the phage to excise. So then we decided to try to follow this a little bit in time with the plate reader experiments. So I'm showing you here in gray the wild type without the reporters, and then we have SPSM MK transcriptional reporter and SPRB transcriptional reporter, either in red or purple. And you can see that around 15 hours post growth, post inoculation, the signal starts being expressed, and we can see a clear increase in fluorescence. So I'm showing you here fluorescence, which was normalized by the OD. So we can see that there is more fluorescence per cell. And similarly, if we observe this under the microscope, so we take different time points, you can see that here at 4 hours, we might have a cell that is fluorescent, which is still originating from the inoculated stock. But then at 8 hours, you almost don't have any sporulating cells, so also not any signal. And then at 16 hours and on, we get a lot of cells which are expressing the SPSM and are therefore giving a red signal. Just to also show you this, how this happens in time, I have here a time lapse. So you can see that here are 2 cells starting to sporulate. Then the spore is being maturizing. And towards the end of this process, you can also see that the SPSM is being expressed and is then really nicely localized in the mother cell. Okay. Oops. Then we were also wondering what happens if we test this in another medium. So since this happens usually when sporulation starts, we try to see if we can try to kind of trigger this earlier if we use sporulation media. So here I'm showing you results for sporulation media. And indeed, the 2 curves for two transcriptional reporters started to kind of separate from the wild type curve around 8 hours. So this started happening before, and it seems that we can kind of trigger sporulation earlier. We will also see this kind of expression earlier. And then we had another question. So basically, how could this lysogenization affect SPSM expression? So for this, we were using 2 different strains. One was lysogenic strain, so P9B1 carrying SP beta, and one was the one that didn't carry this, this phage. Basically, this would mean we wanted to see how the actual presence of this phage could affect expression of the gene that it's interrupting. So to do this, we again had a reporter, either having like both reporters were present in the lysogenic or non-lysogenic strain. And then I'm also showing the 2 controls without the reporters, which are in gray. And as you can see, the fluorescence, the normalized fluorescence, either in lysogenic or non-lysogenic strain, increases gradually. Maybe it's a bit higher in the lysogenic strain, but it seems to be still quite similar. And similarly, if we look under the microscope, we see similar to before, around 16 hours, there are some cells becoming fluorescence, and then at 24 hours, we have more strains being fluorescence. In the non-lysogenic strain, though, here we observed a little bit of difference, like if the cells would start to lyse before and we would have the signal lost. So there might be some differences, but we decided to test this also in sporulation media. And actually for sporulation media, we couldn't observe much difference. So we had, again, nicely separating fluorescence around 10 hours. And then we couldn't really see the difference between the lysogenic and non-lysogenic strain, meaning that actually this action of the phage, excising, it's enabling the bacteria to behave completely normally, like if it would not have the phage. So just to sum up, I showed you today that sp beta phage RS excision takes place during the stationary phase when the cells start to sporulate, and it happens in approximately 5% of cells, which is most probably attributed to the fact that not all the cells start sporulating at the same time, so they gradually, more and more cells are starting to sporulate. So fluorescent phage switching reporters confirmed the expression of attachment gene SPSM and SPRB excisiogenase only in the mother cell during this process. Then also the fluorescent phage switching reporters allowed us to follow this excision at the level of population with the plate reader experiments, as well as in the single cells. And if different media could affect this excision dynamics. And then also expression of SPSM seems to be comparable between lysogenic and non-lysogenic strains, meaning that phage doesn't really affect strongly the lysogenized strain. So in the future, now I will use these reporters to test many more environmental conditions to identify new ones that could trigger this process. And then this will be also used to identify genetic factors, which are either phage-borne or bacterial, to see how the regulation from both sides comes together to regulate this process. And even though this is very fundamental research, it might be of use because this kind of behavior has been observed also in pathogens, and it's also important in different sporulating bacteria, and could help us to control bacteria in a way in medical settings as well as in biotechnological applications. So with this, I would just like to acknowledge the lab, so Dragos Lab and my colleagues, the funding agencies, and you for your attention. Thanks.